I'm going to talk about using language models for computational toxicology. So, okay. So the idea is that toxicology is like a fundamental part of the pipeline of medical research, and is basically the field of biomedical studies that is worried about negative effect of external compounds into biological systems. Typically, we worry about what happens to humans, but there is a classical focus on using animal models in order to derive these predictions. So the idea of computational toxicology is to try to reduce the massive amount of experiments that are needed in order to get these estimates. And to make an example, I have nothing to do with it, but the results I'm going to show you are going to be about require the sacrifice of more than 20,000 rats. So the amount of kills that we do is quite absurd. And from my perspective, given that we might end up in a half a century on the other side of the experimental bench, it's good to start putting out philosophies where this is a stupid idea to do. So the idea is that I will try to drive you through this experiment where we end up using large language model for predicting experimental outcomes from the experimental definition. And it's kind of working. Okay, so this is kind of how this is a huge, TG-GATE is a huge Japanese data set. Now it has more than 10 years old, but it's still the state of the art for multi-compound, multi-measurement studies for toxicology. So the idea is that the experiment has a string defining it, which is basically defining which compound is the rat subjected to, for how long, and with what dosage. Nothing else. But then we have 3 dosage amounts for periods, and 170, at the end we are going to use 150 different compounds. If you think of it from an old school psychometrics perspective, where you're using like ANOVA or over a factorial design, the factorial design here is huge, right? It's 170 by 4 by 12. So of course we are going to be using machine learning models. And this is just the input space. But now, this is all that happens in the output space. We have data about the body weight, we have data about how much food has been eaten by the animal, and then fundamentally we have liver and kidney slides. So these are microscopic slides of the liver and the kidneys that have been done on the rat after killing the rat, sacrificing the rat, after the number of days of its experimental definition. Then humans watched those slides and made annotation of the lesions. So why, in general, why liver and kidney? Because those are the first organs that are processing external compounds before they enter the bloodstream and the rest of the body. Hence, they are very sensible to external lesions. And especially the liver has some crucial necrosis that are very studied. So there are some areas of the liver where most of the blood irradiates the liver, and that is where a lot of lesions are easy to spot. So it's a very good place to study toxicology. Then there are a lot of other data, but we're actually just going to be going over this pathway and ignore the actual images that are 40,000 by 40,000 pixels and worry only about the lesion annotation that comes out of it. I will maybe I do like this. Okay. So the core idea is that then how do we represent experimental conditions? Maybe, but how do we represent compounds? Compounds are very geometrical structures, and compounds are like molecules over a broader, somehow vague definition. And in general, the idea is that there is going to be like a smile representation. Smiles are text representation for compounds. Who was here last month, so some presentation on this from Francesco. And there are these three main approaches that we're going to explore in the talk. QSR, which are traditional quantitative methods from computational chemistry, which characterize a compound based on its structural properties. So these measures are agnostic of what this compound is used with. Then we have deep learning models, and here we have deep learning models specifically trained for compounds. So these models are trained on sequences of smiles, and they are not generally pre-trained language model in the way we know. And then finally, large language models, which, spoiler, are going to work better. Because here we're interested not only in what is the chemistry of this compound, but how does this compound interact with a living creature. And this is a very complex dynamical system problem, and information about this are typically found distributed across the literature and in the discussion part of the papers and not the results. Yeah. So pretty much what is going to happen is that just still go around other 15 times about what the dataset is and the type of approaches. We're going to be mostly focusing early on supervised classification using embedding models. So we will exploit these pre-trained transformers for toxicity and compound representation as inputs for supervised classification model. And then in the final part, we are going to be doing zero-shot classification with large language model with structural generation. So these are pretty much some more numerical definition of the scale of the dataset. The dataset has a lot of different setups. Here we are interested only when the compound is repeatedly given to the rat. So we kind of halve the dataset and we end up with around 15,000 rats. And then these rats are organized in cages. Each cage has 5 rats. And we're going to be studying the cage level dynamics and whether at the cage level we end up having lesion based on the experimental condition. So we have basically this 2000 condition with an average of 5 rats. We look at the label. If the majority of the label from the human doctors are presence of lesion, then we label the experiment outcome as toxic, otherwise not toxic. This is again the pipeline. These are sort of the images, only that they're 50K by 50K pixels. So when you try to put this into some deep learning algorithm, you're gonna require a few terabytes of space just to hold the images. And the humans are actually looking them at max resolution in order to look at cell level instance. Okay, so our objective, start from experimental condition. Humans derive lesion annotation through the liver slide and kidney slides, which are high-resolution microscope images, and our objective is to skip the mapping below and directly learn the mapping above. Again. So here there is pretty much the training setup and evaluation. How does it work? We have 2 separate setups. The first one is what I will call validation scores. And the idea is that, of course, we never train and test on the same experimental conditions, but we train and test on experimental conditions coming from the same compounds. So the idea is that if in train set you might have a low dosage for a small amount of time, then at test set you might have the same compound with a high dosage and a long amount of time. But there is still this matching between compounds. And this is more similar to what we typically call generalization in neural networks. Then the second setup, which is the one that is actually clinically relevant and that toxicologists are interested in, is the setup where we train on a subset of compounds and we test on another set of compounds. So, of course, the sample are always different between train and test, but here what happens is that the group that the samples come from is always different. And this is also very interesting because you need to have high-dimensional continuous representation to do this sort of task. Traditional categorical variables cannot work like this because if you were to learn a categorical model where a compound is a one-hot variable, over a train set, you obviously cannot use this model on a new set of compounds where you do not have an embedding for the one-hot variable. So you need to have some sort of pre-trained or projection method in place in order to do it. So the first approach that we use to regularize the training from the sampling perspective is stratified cross-validation. So when we create the samples for training the machine learning classifiers, we are going to be keeping dosage and sacrifice period and the compounds used in a balanced way, which means that if in the train set there are going to be 80%, if in the train set there are 80% of the sample, 80% of the combination for one compound are going to be in train, 40% in test, and so on. And the idea is, this is the representation of what happens, right? We separate the data set into groups, we sample those groups into train folds, and the validation fold have the same representation of the data. Then the other setup, the one that we're interested in, which is kind of comes from finance actually, where it is used to avoid temporal overfitting. So the idea in finance is you want to train models to predict time series, but you never want to use the future information, right? So the future information are always a block that is never going to be in the train set if you have past information in the train set. So here, for us, the purge is compound. So the idea is that we are never going to be seeing the same compound in train and test. And the way we train this is actually with a two-level cross-validation, where in the inner loop, we train on a subset of compounds and separate them and do this stratification, while in the outer loop, which is our actual evaluation loop, we do the out-of-sample predictions. This allows us to get the score for both setups and to have an unbiased evaluation where we get the same number of prediction over the test set, and we can compute distribution at the sample level through cross-validation. This is, again, I will speed up because I'm almost done. So this is what we call outer purge, inner stratified loop. And, okay, sorry for skipping. How do we, let me jump directly into the results, given that we have 3 minutes. The idea is that, as we said, we embed the experimental context, what is the dosage, what is the time point, and then we embed the compound. So we say that we use text models for embedding the experimental condition, and here we use text and not numerical, like 3 days, 4 days, because these are very nonlinear relationships. We do not have even sampling of days or dosages, but we have this non-ordinal relationship. So we use a language model for this, which is also very helpful. Okay, so then we compare the representation of the compound name between text-based, OpenAI large, molecular baselines, either using Molformer, which is this pre-trained transformer, or using models. This is the old school approaches. The spoiler is that text embedding works well. Otherwise, I wouldn't be here doing the talk. But the idea is that fully textual representation has a slightly better predictive accuracy. And here you can see that validation and test have offset, which is the offset between testing on compounds that you know in new conditions and with respect to testing on never seen compounds. We are still way above the chance level, so it's very high. I am very happy. Okay, so this is the effect where you can see Pareto dominance from the best model. So above, so these are all the different test set that we sampled. So we sampled at the end 1,500 models, and each one of these test sets has 5 different models. So what happens here is that we are ranking the test sets based on their MCC, which is a normalized measure of accuracy. And you can see that there are some test sets where we have 0.25 MCC, and other where we go towards 0.5. And the idea here is some compounds are more difficult to predict than others. So if you have to predict acetaminophen, everybody in the room probably knows that if you take a low amount, it's fine. If you take too much, it's going to be bad. Like your mom has been telling to everybody, asking for a tachypyrina, right? Or something like that. But there are other things that are much more ambiguous, and you can, if you trust me, the post-hoc studies is that cancer drugs that have been released in the last 10 years are the ones that are more unknown, because these have no publication around it, except the publication on them, and they're typically quite private, and they're used only in one experimental or a few experimental trials. So the idea is that this left tails is heavily represented by experimental cancer drugs. Okay, just to conclude my not as clean system prompt. I had to break the markdown in order to somehow show it. So that was with with a combination of embedding models and random forest. And now we are instead going to be seeing using language models. Language models can definitely not do 1,000 cross validations because I'm still kind of averagely rich and cannot do that. So the system prompt is actually very long. This is the whole system prompt. So this has been done together with the FDA, which are partners in this research. So there is a lot of human knowledge on how this is supposed to be done, and a lot of, let's say, PhD syllabus on what toxicity is going through all of it. Like there is some sort of prompting the reasoning structure of what has to happen. And then this is the structural generation schema. So at the end, the model needs to output these variables, and the model is going to be outputting all the variables for a compound together, again, mostly to save money. These are the results. So here we are using the prompt that I showed you, and these are all the models we tested. You see pretty much good models stay on this 67% range, and bad models, or very tiny models, go below it. And thankfully, like these two Quen models actually outperforms, which are considered the best open source models, are actually outperforming the self-supervised, the supervised trained models, and they outperform the big OpenAI models. Just a consideration here, I tuned the prompt and the schema for GPT-4o Mini, and then applied it to all the others. So it's expected that with some graduate student descent, all of the other models are going to be improving as well. And yeah, then we simplify, like we have two ablation conditions. In framework, we do not mention the dataset name, because I would say this is a kind of famous dataset, famous for toxicity purposes, and the model doesn't really have any difference when you do not use the name. And vanilla instead is like something like, you are an expert toxicologist, classify this, like the laziest possible system prompt. And you see that the performance drastically decreased for that one. And I think we are done. These are the last, this is again, compound level data for Quen. So when you see full color, everything is correct. When you see an empty spot, it is a mistake. When you see something like this, it's like 50% of the time. Sorry, I didn't mention that we repeated the experiment 10 times for each language model, and model was very happy about that. And yeah, and so these are just, this is the end.